Class, welcome back. Lesson plan for Thursday. It's going to be viruses today. We do have a couple of things I might send to you for worksheets. I'm kind of kicking around some ideas of how we can get these things back. A lot of them will probably be kind of, sort of the deal where you do them, send them back with a picture, or where they're optional, but strongly encouraged because they're going to be on your test and honor system kind of things. So be looking out for that. Today we do actually have chapel as a fun note. It's probably not going to be right at 9:30 in the morning like it normally is, but we will have chapel recorded and posted to the Rapid City Christian YouTube page, which is where you go to watch like live stream sports and stuff, and where we live stream chapel anyway. So kind of an odd note there, but I think it's cool that we're having chapel with an empty school. So that's worth noting, right? Anyways, we're going to talk about viruses today. The other thing about today that you should know, pardon me, is that I'm doing my first Zoom meeting class. It's like Skype essentially with my anatomy class today. That's about 15 kids and they're pretty tech savvy, so I think we're going to give that a shot. And if that works, I'm going to let you know today. And I'll put a discussion thread here on Edmodo that says what time works best for you for a Zoom meeting. And essentially what we'll do is we'll have class on a Zoom meeting. The whole Zoom meeting will be recorded. And after class is over, I'll post the recording to Edmodo. So be aware that if we decide, the best time for a Zoom meeting is at 1:30 in the afternoon, don't freak out if you get up at 8:30 and there's no biology lecture on at 8:30, right? If we're doing the Zoom meeting at 1:30, then your lecture is going to be posted at 2:30 or whatever. So just kind of food for thought. I think we might give that a shot. I'll let you know how it goes today in anatomy. If it's a train wreck, we won't, but let's give it a shot. So anyways, Chapter 10, here we go. We did bacteria. We talked all about their shapes and their structures, and we did a lab with them and all of this kind of cool stuff. And now what we're going to look at is viruses, which I'm sure you haven't heard anything about recently in the news or social media or anything like that. Nobody cares about viruses right now, right? So viruses it is. Here we go. The discovery of viruses. You don't have to memorize this guy's name nor the date, but understand that our first, let's get a pen here. It was in the 1800s where a Russian biologist was searching for the cause of tobacco mosaic disease. And if you know the word mosaic, mosaic is like a lot of little tiles of different colors, but they come together to make one big picture. And tobacco mosaic disease, basically the leaves had little patches all over them of discoloration. And because it was a valuable agricultural crop, much like most of the research in South Dakota is around corn, soybeans, or pheasants, you research where the money is, right? And so tobacco was a valuable crop, so the research was done on tobacco. And they wanted to know why the tobacco plant was dying and how they could keep this tobacco plant alive. And so Dmitri took these leaves and liquefied them and poured them through a very, very, very fine porcelain filter, like so small that it would stop cells. And at this time, we knew a little bit about bacteria, but we didn't know anything about viruses. And what basically happened was he got the fluid out the other side of this, he got the fluid out of the other side of this filter and knew that that filter had removed cells. So there were no bacteria in the fluid. He took the fluid, poured it onto healthy tobacco leaves, and the leaves got sick. So he knew that the bacterial cells weren't making the disease sick. So what he thought was, well, we know bacteria exist. So the bacteria make a liquid. They make a poison. And when I pour it through the filter, the bacterial cells are held back. But the poison they make passes through the filter. So then, obviously, the bacteria are excreting a liquid that is causing this disease. And so they took the Latin word for slimy poison, which was virus, and they called that by that name. And not knowing that we had actually had the wrong explanation, but the virus word stuck. Of course, it was a virus. It was a virus that's many, many times smaller than a cell and is structured very differently from a cell. And we're going to talk about that. 1946, which bear in mind that's quite a long time later, a gentleman by the name of Wendell Stanley actually isolated the tobacco mosaic virus as a crystal, which is kind of a cool side note, but not something that needs to go into your long-term memory. So let's talk about what a virus is. Viruses are incredibly simple, super simple structures. Basically, they have a core. They have a DNA in the middle of the virus, or RNA, to our knowledge, that we've discovered yet. We haven't seen one with both. So I guess keep your ear to the ground for that, because that'd be kind of cool. And then they have what's called a capsid. Now, write that down, make a Quizlet flashcard, do what you will with that, because in the last section we learned that bacteria have capsules. Now we're saying viruses have a capsid. Okay, so don't get confused with that, because they do sound a lot alike. And a capsid is literally their protein shell. Every virus has a different shape of their protein shell, and their shape largely dictates how they're going to be spread and infect other organisms. And the proteins on the outside of this have to essentially match the proteins inside of you, right? So a virus that infects, the conjunctiva of your eye is going to have different shaped proteins than a virus that infects your lungs, if that kind of makes sense, than a virus that infects your skin, right? So there's the goal here, like the goal with anything in biology, is biodiversity. The more diverse the virus, the more likely it is to dock to many different hosts' skin, or many different hosts' lungs, or what have you. So, some viruses do have a little lipid envelope around the capsid, but not all of them have that. Then they have these glycoproteins that extend out from the membrane, and that's the really, really important part. The glycoproteins are like basically the key. If the virus had a key and your cells had a lock, right? Their key must fit your lock to be able to infect the cell. This is why genetic diversity is also good on the side of the hosts, right? Your cells and my cells have a great degree of variation in the antibodies we have in our blood and the surface proteins on the shape of our cells. And the fact that we're not all identical is hugely important when it comes to resisting diseases. And so it is, in certain senses, a little competition between who can be more diverse, and how can a virus have a key that fits as many locks as possible, right? So the glycoproteins extend from the membrane, they identify the virus, and facilitate attachment to the host cell. So these are how viruses grab on to you. You are the host if we're talking about influenza or coronavirus or whatever kind of virus we're talking about, right? And they also identify the viruses. And so when you go to make your antibodies, sorry, I wish I could write better on this. I apologize for how slow this is. When you go to make your antibodies because you get sick with something and you survive it, of course, because you're young and healthy, then you make antibodies against that virus. And those antibodies are going to be in the shape of these glycoproteins. So that the next time somebody coughs this virus on you, or you get it, you touch this virus, your immune system can rapidly grab onto the virus and say, this guy doesn't belong here. White blood cells, come eat this dude. Let's just play it safe. And that works out really well. So that's antibodies and why they're so important for you. Another weird word, write it down, make a flashcard. We call a virus, and that's it. Technically, when you get viral DNA being made in a host cell, and you encapsulate that with a viral protein, and that thing is a grown-up adult virus, if you will, ready to go and get the world, that's called a virion. Okay, a virion is a complete virus unit. And viruses, bear in mind, we should add this, viruses are not alive, okay? Bacteria are living. Viruses are not living, because to be alive, you have to do a certain number of things. Respond to stimuli, reproduce, those sorts of things. Viruses cannot reproduce without a host. So they're not technically living. If a virus is on your table, it's not growing, responding to stimuli, reproducing the way that bacteria would. It just sits there and hopes to be picked up by a host that it can dock to. And if it dies, then it dies, but it's not going to reproduce unless it's in a host. So without a host, viruses cannot reproduce. And that's the takeaway here. To make new viruses, you have to make new DNA and new proteins. Put the DNA in the proteins, and you're done. Viruses don't have the mechanisms to make proteins. They have no ribosomes. They have no smooth or rough ER. There's nothing in them but DNA. They're so simple. So a virus attacks your cell, puts its DNA in your cell, and then uses your cell's machinery to make more viral DNA and more viral proteins. And this little bitty, epithelial cell in your lungs is becoming a virus factory. And instead of making proteins that are going to make your cell do its job of giving oxygen to your blood, it is instead making viral proteins until the cell is full of viral proteins. Then the cell ruptures and all the baby viruses, virions, come out and attach to the cells next to it. And I have a video that I'll post with this. It's an animation of that process, and it's really quite helpful. So, viruses do not move or reproduce on their own. That's why they're not alive. We call these obligate intracellular parasites, okay? Obligate, you have to. What do you have to do? Be a parasite. You have to attack something else that's living, like a leech, right? And how do you do this? Intra, within, cellular, a cell. You absolutely have to parasitize the interior of a cell, okay? And then your host, be that host a fish or a monkey or a person or a bacterium, will carry you to other fish or monkeys or humans or bacteria and give you to those other organisms. And then they all get sick, and on and on and on this process goes. So that's the goal. All right, let's talk about shapes. The shape is determined by basically what your DNA or RNA, your nucleic acid, is going to code for a protein. That protein makes you the shape you are, right? And viruses come in a lot of different shapes. I don't care that you memorize like which shape goes with which virus. Just get the big picture here, right? Tobacco mosaic virus is a big helix, right? It has 20 triangular sides, and it looks like this. This is kind of a cool deal, right? 20 sides, and it's a helix. It's all wound up like DNA is. And so here is your RNA core, right? And the core is going to be DNA or RNA. In tobacco mosaic virus, it's RNA. The outside is protein. All these proteins are the same. Those proteins allow it to dock to the cells on a tobacco leaf. But those proteins are the wrong shape to dock to the cells of a person's skin. So the person can handle tobacco mosaic and work with it in the lab, but they don't start growing weird patches of off colors on their skin because this guy is specialized to attack plants. Does that kind of make sense? I really miss the opportunity for you guys to ask questions, so it's a tough thing to lecture in this manner sometimes. HIV looks like a gross soccer ball. Again, we can't see the DNA or RNA in this example because all we're seeing is called the what? The capsid, okay? So this is a really cool idea because the capsid is this guy here. We're just going to write cap for capsid and save ourselves some time. The capsid is this green soccer ball, right? And the glycoproteins, which are how it docks to the cells, and in the case of HIV, it's white blood cells, which is just terribly ironic because bear in mind that white blood cells are the cells which find and kill the viruses. So to have a virus that gets inside of white blood cells is sort of like having a bad guy take over the security guard, right? And put on the security guard outfit. And that's not great because how are you going to kill a virus if it's in your immune cells, which is why HIV is a lifetime virus and you will never, ever, ever get rid of it. So the glycoproteins are these little hairy donuts, if you will, kind of a gross picture. That's what actually allows HIV to dock to your cells, okay? So that's an important thing, capsid and glycoprotein. And so that's HIV, human immunodeficiency virus. Is that right? HIV? Off the top of my head, I'm not quite sure what it is. There's HIV AIDS, right? HIV is the virus. AIDS is the disease you get later, which I think is acquired immunodeficiency syndrome. But don't quote me on that. This is just kind of off the top of my head. I think that's what it is. And then Ebola. A few years ago, before coronavirus took over the news, there was a disease called Ebola that came out of Africa and had people super freaked out because the mortality rate was high. like high, like 90%. You're gonna die high, and people were scared. What we found was that well, basically we did a pretty good job quarantining it. I think only like 2 cases came into the United States, and what we found was that those guys weren't dying. And in developed countries where healthcare was pretty good, people weren't really dying, which was an interesting thing and also a sad thing because it spoke to the terrible healthcare conditions in the regions of Africa where folks were like literally just laying in the muddy alley and dying, and that was very sad. And it was also visually a little bit of a scary virus as well. But I'm sure you guys don't even remember that because you were like I don't know 12 or something like that, and probably didn't care. And so here's Ebola. It's a cylinder. The cylinder is all like curly like this, and so basically this is a two-dimensional picture, so it's a little tricky, but just imagine that this is a tube, like PVC, right? Bent, and that this white space here is the hollow interior that has the nucleic acid, DNA or RNA, and then the protein is what makes it a cylinder, and that's Ebola. This picture is also famous because it's the picture from an old movie in the 90s, which again, you guys wouldn't probably know, but it was called Outbreak, and it's a horror movie, right? A science fiction, a thriller, whatever you want to call it, basically a disease that comes out of Africa that takes over, the nation essentially, and it's this big race to try to stop it. And you guys might be familiar with the more recent Contagion, which was from the 2000s. You guys would have been a little older then, but I thought it was so interesting that they took a hypothetical virus to make their movie that came out of Africa and had a super high mortality rate, and then they used the picture of Ebola to show the hypothetical virus. And I don't know if that irony escaped a lot of people, but I was like, wow, that's holy smokes. So I thought that was kind of, I don't know if funny is the word to use, but it was it was something. So anyways, how do we classify our viruses? Basically, you ask yourself, is it a DNA virus or is it an RNA virus? That's a great way to start because the protein shapes are so diverse. We can't say, well, this is the icosahedral virus and it's in the family of icos. No, that's crazy. Is it DNA or is it RNA? And how does it infect its host? And that's basically the easiest way to do it because we can test their DNA or RNA. Even in a lab setting, you can get viral screens done. They're quite expensive and they're not that popular because you have to basically check all the DNA in a person's saliva against the logged DNA of a host of viruses. And the reason they're not incredibly popular is because viruses, which this is the part that frustrates people, I think, is if you get sick with a bacterial disease, we give you an antibiotic. The antibiotic kills the bacterium and bacteria, there'll be more than one, and then you're in good shape because the antibiotic helped you. Viruses, once they're in you, have to run their course, right? And that really frustrates people. But basically, this is symptomatic care, right? They're hot, try to bring their fever down. They're dehydrated, get them to drink fluids. They can't drink fluids, hook them up to an IV and hydrate them that way. They're struggling to breathe, put them on a ventilator and wait. Your immune system must basically win on its own. There are a handful of viral drugs, and we'll go over some of them, and there's probably more now, but that's generally the rule, is your immune system has to beat it, and that makes people very frustrated. So our DNA containing virus, this you have to kind of go back to chapter 3, which was cells and how cells work, to understand how viruses work, and it's really not that hard. But basically here's what happens. You guys know already that the central dogma of molecular biology is that DNA is turned into RNA, and RNA is turned into protein, right? Every kid who comes out of any biology class has to know this, right? This is like biology for babies. DNA to RNA, RNA to protein. The DNA to RNA is called transcription, and the RNA to protein is called translation. In the nucleus, we turn DNA into messenger RNA, or mRNA. mRNA wiggles his little self out of the nuclear pores in the nuclear envelope from chapter 3 and will be found by a ribosome. If it's a free-floating ribosome or it's attached to the rough ER, I don't really care. And neither does the RNA. But the ribosome grabs that RNA and turns it into a protein. And then the protein, if it's folded in the right shape, goes to do its job to make your hair curly, or produce melanin to make you tan, or be an antibody to try to fight off influenza or whatever you just got vaccinated for. It doesn't matter. Proteins do everything. If you don't know, guess protein, right? You're probably going to be close. So, what does this mean for a virus? Okay, the virus docks on you. Let's say it docks on you. It's more exciting than to say the virus docks on a plant leaf. Nobody cares about plant leaves, right? Viral DNA is shot into your cell, okay? Now, your enzymes will grab that DNA and turn it into RNA. You will do this step for the virus. You will do that for it. And that RNA will then become protein. But instead of making the proteins that are going to make your cell do its job, whether that's a lung cell or a skin cell or whatever, right? You're now making the capsid. Your cell has been turned into a factory that is going to produce capsids. And you will copy the viral DNA just like you copy your own DNA. And you'll make more viral DNA and more capsids. And we'll put the DNA in the capsids, and your cell will be filled to bursting with virions. Then your cell will burst, and those virions will dock to your neighboring cells and take over those cells. This is the process of infection. Okay? So we're actually going to stop there because I do want you to watch this influenza animation. I will put this in the description. Okay? Every day is an Edmodo post, right? I will put this in the description of your Edmodo post. And so you don't have to try to type in all of this nonsense, and you'll click it and it'll take you to YouTube. So today, watch this guy, the lecture that we're just wrapping up now, watch the influenza animation, and we're going to stop there. Next time we're going to try for a Zoom meeting, so go ahead and reply with the time that works best for you, and we will see if we can get a few people together at least, so you can ask questions, because right now it's just me talking, and you don't have the ability to ask questions, and I'm sure that since you're literally all home right now, quarantined, due to a virus, there's got to be at least one kid with one question that I'm missing. So anyways, as always, thanks for your patience. Thanks for your hard work. 4 of you have already turned in your lab report, and it's not even Friday, so I think you guys are doing awesome. If you have any questions, don't be shy. Email, Edmodo message, drop me a text. I don't care. We're going to have a fine time. You guys are still going to learn a lot, and we just got to get this dialogue going a little smoother, because right now it's a monologue at 5 a.m. in my kitchen, and that's weird. So I have to get you guys plugged in somehow. Anyways, I sure appreciate you guys' time, and I'll talk to you next time. Thanks.